Sathyabama Institute of Science and Technology.
Hello students, this is Dr. Indhumathi from the Department of Biotechnology, School of Bio and Chemical Engineering, Sathyabama Institute of Science and Technology. Today I am here for a presentation on the topic Mechanism of Eukaryotic Translation. Introducing the topic, translation is basically a process where the genetic code or the genetic information contained within the mRNA sequence is actually translated to form a sequence of amino acids to join the protein synthesis. This basically happens in the cytoplasm. That is, eukaryotic translation happens in the cytoplasm and it follows three stages: the initiation, elongation and termination. Translation is also called as protein synthesis because the sequence of amino acids join together to form a final functional product called the protein. And there are certain essential components that are required for the synthesis of proteins, and they are the amino acids, ribosomes, mRNA, tRNA, protein sources and energy sources. Amino acids, we basically know that there are 20 amino acids and 10 are essential amino acids, which means they have to be acquired only through the dietary supplement, and other 10 amino acids are synthesized by our own body. So there should be all the 20 amino acids present for a protein synthesis to happen. Coming to the ribosomes, so ribosomes are also called as protein factories, or the factories where protein synthesis is happening, or you can also call it as the workbenches of translation. Ribosomes of eukaryotes are basically of 80S type, which has a larger and smaller subunits, that is the 60S and 40S subunits. And ribosomes in eukaryotes have three sites, basically P site, A site and another site called E site, called the exit site. Ribosomes always couple with the rough endoplasmic reticulum where the protein synthesis happens, and ribosomes are always seen in the cytosomal fraction of a cell. Then we have the mRNA. The mRNA actually contains the genetic information that has to be translated into the proteins. We have tRNA. tRNA, which basically has 5 arms: the acceptor arm, anticodon arm, T-Cy arm, DHU arm, and the smaller one, the smallest arm, which is present on the right side near the DHU arm. The two major arms that are involved in protein synthesis are the acceptor arm and the anticodon arm. Anticodon arm actually has 3 codons usually, which will actually pair with the mRNA sequence if it is complementary to it. And the acceptor arm usually has the activated amino acids, and this activated tRNA will donate its amino acid to the growing polypeptide chain. And we have energy sources like ATP and GTP that are required during certain steps during protein synthesis, and we have protein sources. The protein sources majorly involved here are the initiation factors, elongation factors and release factors. So before the protein synthesis could begin, a particular step called activation of amino acids must happen. So in the process of activation of amino acids, a group of enzymes called amino acyl tRNA synthetases are involved. These enzymes are very particular to a particular amino acid actually. So if this enzyme is going to transfer methionine, this enzyme will be called as methionine tRNA synthetase. If it is going to transfer proline, it will be called as prolyl tRNA synthetases. If it is valine, it is going to be called as valyl tRNA synthetases. Usually the amino acid makes a complex with ATP along with a particular enzyme, and this complex is usually transferred to the acceptor arm of the tRNA, that is the 3' end of the cloverleaf structure of tRNA, and this tRNA is an activated tRNA, or you can also call it as amino acyl tRNA, which contains an activated amino acid, and this process is called activation of amino acids. Coming to the major steps in translation, the initiation, elongation and termination. First we'll look into what is initiation. Initiation is a very complex process in eukaryotes because it involves at least 10 initiation factors. We call EIF, E for eukaryote and IF for initiation factor, and the process of initiation is actually divided into 4 basic steps again: the ribosomal dissociation, formation of 43S pre-initiation complex, formation of 48S initiation complex, and formation of 80S complex. So first we'll look into this dissociation, ribosomal dissociation, where the 80S ribosome will actually get dissociated into its 2 subunits, the 60 and 40S. As you can see here, this is a diagrammatic representation of the process of initiation, where the 80S ribosome will split into 2 subunits, 60 and 40S. To the smaller subunit 40S, initiation factors like IF3, IF1A and IF5 get bound. This complex will actually form a structure called 43S ribosome. To this 43S complex, we have another complex called the ternary complex. The ternary complex include the first tRNA, that's called the methionine tRNA, which means this tRNA will actually have the amino acid methionine in its acceptor arm. So this methionine tRNA, that's met tRNA plus the initiation factor 2 and GTP. So this is a complex and this is called ternary complex. This ternary complex joins this 43S ribosomal complex and it finally forms the 43S pre-initiation complex. In this process, the GTP is hydrolyzed into GDP. Energy is required here, followed by the formation of 43S pre-initiation complex. You can see the 43S pre-initiation complex here, where you have the 40S smaller subunit to which the initiation factors 1A, 3, 2 and the initiation factor 5 is also bound, and this entire complex is called the 43S pre-initiation complex. To the 43S pre-initiation complex, we have our mRNA which is ready to be bound. This mRNA actually makes a complex with another initiation factor called EIF4F. This 4F is again a complex of 3 more initiation factors, 4A, 4G and 4E. This 4F is basically a cap binding protein which actually recognizes the 5' capping of the target mRNA and binds to it. So this mRNA is now bound to the initiation factor 4F, and this complex is now ready to join or to make the 48S initiation complex. At this time, the ribosome actually scans the mRNA for the presence of the initiation codon. The ribosome wants to know where the initiation codon is placed in the mRNA. To identify the location of the initiation codon AUG, the ribosome will scan for certain set of sequences that exists before the start codon, and this particular sequence is called as Kozak sequence in eukaryotes, and the same sequence which is used for recognizing the initiation codon is called as Shine-Dalgarno sequence in prokaryotes. Once the ribosome scans the entire mRNA for the presence of Kozak sequence, it will position the mRNA in such a way that it starts with the initiation codon AUG. So now the mRNA which has been scanned by the ribosome and it has already complexed with initiation factor 4F joins the 43S initiation complex and forms the 48S initiation complex, and during this time an ATP is hydrolyzed to form ADP and AMP. You can see here the 48S complex where the 43S complex is already present, to which the mRNA which has a 5' capping is joined and it starts with the initiation codon AUG, and this entire setup makes the 48S initiation complex. This 48S initiation complex further on tries to join with the remaining 60S, the larger subunit of the ribosome, and it makes the 80S initiation complex. During this time, a GTP is hydrolyzed to form GDP, and initiation factors like IF5B is actually joining the process. Once the 80S ribosomal complex or the initiation complex is formed, all the initiation factors like 3, 1A, 4F, 5, 2, everything will be degraded and they are recycled back to another cycle of initiation of translation. And finally, we have this 80S initiation complex which have both the 40S and 60S subunits with the mRNA in between. Followed by initiation, we have the process of elongation. Elongation is actually the sequential addition of amino acids by the ribosome, and this step is a cyclic process because this will be repeated until the required number of amino acids are added, and that will end in a termination process finally. So this process of elongation also is divided into 3 steps. First is the binding of amino acyl tRNA to A site. Next is peptide bond formation, and the last one is translocation. So, in the binding of amino acyl tRNA, I have already spoken about what is an amino acyl tRNA, what is an activated amino acid. So, you can have a look of what is an activated amino acyl tRNA. So, this is an amino acyl tRNA which has an anticodon arm here, and it contains 3 codons that will find its complementarity with the mRNA, and you can see the 3' end of the tRNA that holds the amino acid, and this setup is completely called as an amino acyl tRNA which has an activated amino acid in it, and this plays a major role by adding amino acids one by one to the growing polypeptide chain. So, the process of elongation starts like this. So, as you can see, this is a complete ATS initiation complex which has E site, P site and A site, and an mRNA is positioned in between. So, in the P site, you can actually see the met tRNA, which is the first amino acyl tRNA added to the complex. So, in the P site, this met tRNA is positioned, and the A site of the ribosomal complex is now empty. The empty A site will now attract another amino acyl tRNA. So, we have so many tRNAs in eukaryotes, like nearly 50 different tRNAs, as we have 20 different amino acids, but 64 possible combinations of codons, right? So, for the 64 possible combinations of codons, we need to have different types of tRNA. So, we have nearly 40 to 50 tRNAs in human body itself. So, those tRNAs will try to come into the ribosomal complex and check for the complementarity one by one. So, the tRNA which actually finds a complementarity between its anticodon arm and the mRNA's codon will actually bind to the A site. So, you can see here the P site is already loaded with the met tRNA, A site is empty, another activated tRNA is trying to enter the A site. It checks for the codon complementarity between its anticodon arm as well as the mRNA and tries to join there. Once after joining, you can see here the another second amino acyl tRNA has joined the A site also. Now, in the ribosomal complex, we have 2 amino acids already added, but the next step should be and during this process of binding of amino acyl tRNA, we have the elongation factor 1 that is involved here, and we need a molecule of GTP also, because this process does not need any energy because already the amino acids are activated, so there is no need of any energy. Following the addition of the amino acyl tRNA to the A site, then comes the peptide bond formation. So here an enzyme called peptidyl transferases are involved. This peptidyl transferases activity is relying on the 28 S RNA of the 60S subunit. So this enzyme actually is a ribozyme, which means the ribosome itself acts as an enzyme to form a peptide bond between 2 amino acids. So this enzyme plays a major role in it, and this process requires the help of ATP to be hydrolyzed here. So during peptide bond formation, the enzyme peptidyl transferases actually tries to form a peptide bond between 2 amino acids, adjacent amino acids, which is actually from the P site and A site. So you can clearly see the P site is loaded with the met tRNA, A site is empty. To the A site, the second amino acid tRNA comes and joins, and you have 2 amino acids now without any joining, without any conjoining in between them. Followed by that, we have the peptide bond formation, where there is a peptide bond that is forming between the 2 adjacent amino acids. Once after the peptide bond formation, the next step would be translocation. Translocation is the process where the ribosome starts to move across the mRNA to the next codon, because the next triplet codon has to be read for the addition of next amino acid, and this process requires more energy. So a molecule of GTP or ATP is required here for this movement of mRNA. Ribosome starts to move from the initiation codon and the next codon to the third codon now, and now the initiation codon comes front, which means the mRNA will be pulled front, ribosome will be moving to the next codon, and the P site which already had the met tRNA will be now moved to the E site and it exits via the E site, and the activated tRNA that was in the A site will now move to the P site. So this cycle will be actually repeated. Now the A site will be empty, another tRNA will enter and check for the complementarity of the codon, and then it will add the another amino acid, and this cycle will be actually repeating. So once after the formation of peptide bond, the mRNA translocates, as you can see here. The tRNA which was present in the P site will exit via the E site, and the tRNA which was present in the A site moves to the P site. And finally, the next amino acid tRNA will join the A site to check for the complementarity here and try to join its amino acid. So until the chain of amino acids are fulfilled, this process will be repeated. So this is a cyclic process, as it is already said, and this will finally end in termination. So termination process is a very simple process here, it's not complicated. So what happens during termination is, after the ribosome translocates through the mRNA, and when it has read all the codons and all possible combinations of tRNA has bound to it and contributed its amino acid to the growing polypeptide chain, finally when the mRNA reaches one of its top codons called the UAG, UGA or UAA, these three codons. So when they reach either of these codons, there is no tRNA for any stop codon. So once the ribosome recognizes that this is a stop codon which is present in the mRNA, there is another protein released called the eukaryotic release factor. Not only the eukaryotic release factor is involved here, along with that we have the peptidyl transferase enzymes as well as the GTP molecule involved. So the enzyme, ATP as well as the release factor makes a complex and joins in the ribosomal complex here. Once after the growing polypeptide chain has grown to a required length, and once it has read the stop codon, the ribosome has read the stop codon, this complex, that is the peptidyl transferase, ATP and the release factor all together breaks the peptide bond between the polypeptide chain and the tRNA in the P site, because every tRNA after forming the peptide bond will actually shift to the P site, leaving the A site to be empty for the next tRNA to join, right? So when there is only stop codon, there won't be any tRNA joining in the A site. Now the A site is empty finally. The P site only has a tRNA which contributed the final amino acid to the growing polypeptide chain. This enzyme, GTP and release factor complex will cut the peptide bond between the grown polypeptide chain and the tRNA in the P site, and the tRNA will actually exit via the exit site, and the grown polypeptide chain is also released. The ribosomal complex then dissociates to form the 60S 40S subunits again. Through this process, the mRNA which was stuck in between the complex will also get released, and this marks the end of translation termination. So these are my references which has been taken from biotechnology by Satyanarayana and other research papers. Thank you students for patiently watching the presentation.